obviously we're going to be talking about the vascular tissues. So I'm going to try to like tie it together with the first chapter and review a little bit, so hopefully you know something. So what are the plant tissues? Those are cell types. The type of tissue that xylem and phloem belongs to is vascular.
Dermal and ground tissue?
Yes. Okay, vascular, dermal, and ground. So vascular is xylem and phloem. What's the dermal tissue?
Like
Right, the outside covering of everything, the outside covering of the leaves, the stem, the roots, okay? And then the ground tissue is everything that isn't vascular or dermis, okay? Or dermal. So we have those, and then we have our cell types, which are parenchyma, collenchyma, sclerenchyma, okay? Which one are the normal ones? Single cell wall, not strong. Parenchyma, super strong double cell wall, really, really super thick. Sclerenchyma, and the reason why I bring that up is because which part of the vascular system that we're doing today is sclerenchyma?
vein.
The xylem, okay? The phloem is parenchyma, and you know that because if you remember from our first lesson, we said that when like a tree grows in width, which part is the wood? The xylem. And you know that's super hard. So you know that's going to be the sclerenchyma, and then the bark is part of, or the phloem's part of the bark, and we know that that is parenchyma, and it continues to like slough off throughout the year so that it never gets, like, you don't get great big rings of phloem, you just get rings of xylem, and you only have one ring of phloem, okay? So just kind of put it in perspective, and now we'll get started. I'm going to try to keep tying it back together as we go. Hopefully that's not going to put you guys to sleep. Oh, good. Yes, definitely need that somewhere. Oh my goodness. I think it's the 19th now. 14th was Sunday.
Yeah.
Okay. So guys, there's basically three kinds of transport that a plant does. If you think about transport in us, our vascular system, we know that it's our blood, and we just have blood. Everything is carried through the blood and then diffuses out, but they have two kinds of vascular. They have xylem and phloem. But in addition to that, we're going to talk about local transport, which we do. You can sit down. Huh? You can sit down if you want.
Is there a seat?
Yeah, there's like a, I won't bite. There's some seats up in front.
6, 7 seats.
There, here, there. Anyway, so we're going to first talk about local transport. Actually, we could skip this, but I thought it might not be a bad idea because it's the same way stuff is locally transported anywhere. So it's just a quick review, okay? Then there's lateral transport, which is kind of close by transport, but not super close. And then we've got long distance transport. Okay. Ooh. Okay, ready? I said we were going to go quick. We're done. Okay. So first thing is going to be facilitated diffusion, and everybody remembers what that is. What defines facilitated diffusion? Okay, high to low and through protein channels. Okay, and this is just a reminder that the channels can be open. They can be, but even if they're open, they're specially shaped. Okay. Then we have shape change, where it bangs into it, it makes it change shape, and then the thing either goes in or out, depending on which side has the higher concentration gradient in the cell or outside of the cell. And then we have the gated channels, which need a signal to open in order for the thing to diffuse. But they're all diffusion. It's all movement of high to low. So I just thought it wouldn't be a bad thing just to remind you of that. But I don't think we need to spend more time on it, do we? Good. All right.
High to low diffusion.
Yeah, and diffusion is always high to low.
You said the second one. And
Protein channels. Okay, then we have active transport, which is also protein channels, but ATP you need for it, and it's right against the gradient low to high. Okay, Actually, there are multiple kinds of active transport, and we're going to say normal, and that is where the ATP activates the channel changes shape and throws stuff out or sucks stuff in. It just depends on the channel which way it goes. But it's driven, the shape change of the channel, rather than being driven like in facilitated diffusion and the shape change there, rather than being driven by the molecule hitting it, changing the shape, the ATP changes the shape. And therefore that ATP is energy that the cell has to make. That's why it's called active transport. That's the difference. So just to reiterate that, it's been months since we did cells. It's been months since we got to school. So I just thought I would mention that. Plants obviously use chemiosmosis in photosynthesis and cellular respiration, but they also use chemiosmosis as a mode of transport. Basically, chemiosmosis is defined, we defined it very specifically in cellular respiration and photosynthesis as what? Right, and the pumping of the hydrogen ions increases the concentration, and then the hydrogen ions themselves flow through the ATP synthase channel, creating ATP. But chemiosmosis technically is any time the pumping of protons drives anything. It doesn't have to drive the synthesis of ATP. It could drive the movement of molecules through a membrane. It's still called chemiosmosis. So I have an example up here, and we're going to come back to it. But basically, it might be better to come back to it. But if we had the epidermis, and if you remember, we also reviewed last time leaf structure. We have to have little openings, and we're going to have guard cells in those. More guard cells where? Top or bottom? Yeah, on the bottom. I just drew one on each though. Okay, and these need to be able to close and open. So if the stomate's open, then stuff can move through it. If they're closed, it can't. When do we want the stomates open? Because what's the purpose of the stomates? We're just going to try to catch various things as we go. Okay, carbon dioxide needs to go in, and oxygen needs to come out. And we don't want it to, but water vapor will come out as well. So that's the idea of the stomates. So when do we want them open? Okay, when? Daytime, okay. Want them open during the day because you can't do photosynthesis at night, so why open them and chance any water evaporation when it doesn't do you any good to get CO2 anyway? Yeah, up there's good. So during the day, okay, when there's plenty of water, because even if it's the day, if all the water is evaporated out of the plant, then we can't photosynthesize anyway. So if we have them open, then we're going to lose all the water out of the plant, which is going to really mess it up, and we're not really doing anything anyway. The other issue is we would like them open when the carbon dioxide concentration goes down. So think of our summers, right? It gets really hot and sometimes really dry. If the stomate's closed all day in response to dryness, there will be no photosynthesis. It can happen, right? Doesn't your grass brown up sometimes during the summer and do nothing? But in good summers where it's hot but it's not totally dry, what's going to happen throughout the day is the stomates are going to open and close. Even though it's the day, they're going to keep opening and closing. Mainly, they're going to open when the CO2 in the leaf gets low. So that makes sense? And then they open, CO2 diffuses in, and then they close again when the CO2 gets high. So basically, there's three controlling mechanisms. So it is the light, it is the CO2, and it is water. All three of those control that. So I mean, I kind of jumped ahead because I'm going to do this yet. But basically, the chemiosmotic part is, I did tell you this already, that what triggers the guard cells to open is when they swell with water. And that makes sense because you want them to open when there's plenty of water. But what happens is potassium actually flows into the guard cells, and as potassium goes in, it makes the inside of the guard cells what? Hypertonic. Water flows in, and I'll show you in a minute why water flowing in makes them open. Okay, I know it doesn't make intuitive sense, but it's basically the weird